Today I'm going to share on the topic cloning process in genetic engineering. So definitions. So cloning is a genetic engineering refers to the process of producing identical copies of DNA fragments, genes or entire organisms. The most common type is gene cloning, which involves creating multiple identical copies of a specific gene or DNA sequence. So types of cloning: molecular cloning or gene cloning. It makes copies of specific genes or DNA segments. And second one, cell cloning, producing identical cells from a single parent cells. And organismal cloning, creating genetically identified organisms, that is a dolly, the sheep, the first one to be cloned. Steps in cloning process: isolation of DNA, that is gene of interest, and selection of suitable vector, insertion of gene into vector, introduction into host cell or introduced into the host cell, selection and screening, cloning and expression, harvesting and analysis. So the isolation of DNA and selection of vectors. The target gene is isolated using restriction endonucleases, and the gene of interest is identified and isolated. Then selection of vectors. Vectors like plasmid, bacteriophages, cosmids, bag and yak are used to carry the genes into host cells. And cutting DNA with restriction enzyme. Restriction endonuclease cut DNA in specific sequences like chewing, sticky or bluntends. Insertion and introduction into the host. Insertion into the vector. The DNA fragment is inserted into a suitable vector, that is plasmid, and ligated using DNA ligase. And the recombinant DNA is introduced into host cells by a transformation method such as heat shock, that is calcium method, electroporation, microinjection and gene gun method. Selection, screening and expression. Selection markers will be antibiotic resistant genes, which is identified by transformed cells. The recombinant DNA is introduced into a host cell, that is usually E. coli is preferred. And the selection and screening, the transformed cells are selected using markers like antibiotic resistance or color change. And the expression, the cloned gene is expressed and its product or presence is verified using molecular techniques. Then harvesting and analysis. The cloned DNA or expressed protein is extracted and purified. Analytical techniques include PCR, gel electrophorations and sequencing. So this is the diagram showing the cells having the gene of interest, and the right side showing the bacterium which is having the plasmid. So these two cells are allowed to cut with the restriction enzymes, and in this the gene of interest from the cells are isolated by using the enzyme, restriction enzyme. And here the bacterium, again the same enzyme was introduced to isolate the plasmid, which shows a cut ends. Then the gene of interest and plasmid are fused together to form a recombinant DNA plasmids, and this is said to be recombinant bacterium. So from this bacterium, many bacterium are cloned. So this is said to be clone of cells which contains a gene of interest. So overview of gene cloning process. First, the donor DNA is taken, and from that the gene of interest was chosen and it is cut with restriction enzymes. Then that vector DNA is cut with the same enzymes on the other side, and the ligation happens with the recombinant DNA. And it is transformed into the host cell what we have chosen, and the selection of recombinant clones or taken, and it is taken for gene expression process, that is by PCR technique it was analyzed. Here, what are all the enzymes used in cloning is restriction endonuclease that will cut the DNA at specific sequences, and DNA ligase it will join DNA fragments, and reverse transcriptase it converts RNA into DNA, that is for cDNA cloning, and that is complementary DNA, and DNA polymerase it amplifies DNA. Then applications of cloning. So production of recombinant proteins such as insulin, growth hormones can be identified by using this cloning process, and gene therapy for correcting defective genes, and development of genetically modified organism, that is in short called as GMOs, and stem cell research and regenerative medicine, conservation of endangered species, that is by reproductive cloning, and biomedical and pharmaceutical research process these applications are carried out. Then advantages. It produces identical copies quickly. It enables large scale gene or protein production, useful for studying gene function, assist in disease treatment research. And limitation, there are some limitations for cloning process. It should be done by getting ethical concerns in organism cloning, and there is a risk of unwanted mutations, and technical challenges in gene expression regulations will be there. And what will be the disadvantages will be the reduced genetic diversity, high failure rate in animal cloning, ethical issues in human or embryo cloning, and possible genetic abnormalities in cloned organisms. So in this cloning, the first sheep, that is called Dolly, in 1996 it was first cloned mammals from an adult somatic cell. So it is created by somatic cell nuclear transfer, that is SCNT. The nucleus from mammary gland cell inserted into nucleated egg cell, an electric shock stimulated division, and embryo implanted into surrogate mother, and the result is Dolly, the genetically identical to donor sheep. So simple process of somatic cell nuclear transfer is donor nucleus plus enucleated egg that gives clone when it is fused with the donor nucleus and nucleated egg, and after fusion the embryo forms, then embryo to next step surrogate, and then next step the cloning occurs.